And then, like since there, I stayed in Boston. I started my lab in 2016, and I've been funded mainly by NAGRI. Today, like I'm running a mid-sized lab, and we focus on gene regulation. So one thing that we like to do in my lab, we really like to build computational methods. So these are some of the methods that we build since I started the lab, and I know that we will have meetings later, so I will not present all these tools. We don't have the time, but just want to give you some pointers. So to drive discussions. So these are some of the tools that we develop. One that I will briefly present is a tool to try to find regions that are important in gene regulation, integrating epigenomic and gene expression data. It's called HStack. We've been also working with single cell data, trying to reconstruct developmental trajectories using gene expression data and chromatin accessibility, and it's a tool called Stream. And then we've been really active in creating tools for genome editing and try to create tools to design perturbation, but as well to quantify perturbation. So CRISPR and CRISPR-Me tool that is not in this slide there to design perturbation. CRISPResso and is to quantify genome editing activity. CRISPR-Surf, I will present today, is a tool to dissect regulatory elements. And Prime Design is a tool for a new technology called prime editing that is a really cool technology to precisely insert insertion, deletion, substitution without double-strand breaks. And we're also excited, to model transcription factors. So we have, two tools that we recently published. One is called Motif Raptor that essentially allows you to understand which factor may be more influenced by genetic variants. So integrate GWAS, chromatin accessibility, and RNA expression, and a tool called Graphemo that is a simple scanning tool like Fimo, but instead of scanning linear genomes, we scan recent graph genomes. And finally, we also really excited about new visualization techniques. So we developed this tool for fun called single cell VR, where if you buy this Google Cardboard for $10, you can see your single cell data set in virtual reality, and you can fly in your UMAP, and it's just a fun website. And these are all published, so I will not spend too much time on all. I would just focus on a few of these tools. So today I have two segments, and in the first segment I will talk about CRISPR genome editing. How can we use these technologies to dissect functional element in the genome? In the second part of the talk, I will present some unpublished work where we use single cell data to build regulatory maps using graph embeddings. So let me start with the first one. So we are really excited about understanding genome function. Despite we have the full sequence of the genome. In 2001 we had the full draft, but it was not until this year that we have the full sequence. But there's still a lot of things that we don't know. Like for example, if you're in the coding space, you understand exon, intron, like all these things are working much better. But if you are in the non-coding space, if I highlight one region that is outside the coding space, sometimes we don't fully understand how these sequences are working. So the big question is like, how can we learn to read and understand this non-coding part of the genome? And one reason why we want to do that is that first of all, 98% of the genome is non-coding, and 80%, depending on which assays you use, as some biochemical activity. We don't know if all of this is functional, there are like debates, but still, there are like some some biochemical activity you measure, but one important thing is that many of the disease-associated variants are in this space. So it's really important to try to understand what's going on in this part of the genome. So one thing that, is also important to think about, if you want to understand the genome, and, to me, it was a puzzle, as a computer scientist, this was one of the reasons why I wanted to transition to computational biology. And, the question is like, how come, our cell have the same DNA, but they do really different things. They activate different programs, but the program is the same, right? So this was really like puzzling to me. And, one thing I learned, during my postdoc is that actually, you have the code, but this code can be activated, organized in different way. And, one of these, the key idea is that the way this DNA is organized in the chromatin is really important. This chromatin is really dynamic, it's not static. So this is partially explain this. And there are like different things that can control the organization of the chromatin, like, the nucleosomes, the DNA methylation and histone modification, and many other things that I don't have in this slide. But so with these things in mind, we know like some of the actors, right, that, may be important in controlling this program and the gene expression, but the genome is still really large, right? So how do we follow, how do we explore this 3 billion base pair in the genome? So what do we need? So one thing that we need is like, we have a way to prioritize, we need to highlight, some region of the genome. And if we really want to understand the function, the best way is to perturb. So, and for this, we have like different gene editing technologies, and this leads us to understand that we can iterate in this paradigm. So to highlight, as I mentioned, there are like multiple ways. One really effective way to highlight important part of the genome is to use, for example, chromatin accessibility or histone modification. So if you have not heard about this concept, the DNA is wrapped around nucleosome, and these have particular tail, and this tail can be modified. And one thing that people have described is that depending on which modification you have in this tail, you can actually potentially infer the function of that part of the genome. For example, if a gene is transcribed, you will see this H3K36 trimethylation, and if the gene is repressed, maybe there are particular mark in the promoter. And there are a lot of marks that people have described. One that is really nice and important in this exploration is this H3K27 acetylation that mark this region that are called enhancers. And these are regions that you will not find easily if you don't have a way to highlight. So these are regions that can control gene expression from distance, and can fine-tune the activation. And many of the variants are actually falling in this region. So it's really important to have a way to map. And thanks to roadmap epigenomics and encode, we have like this tracks for several cell types, and then we can focus our exploration. And for this, my lab has developed this tool that can use this mark or also other mark, this A-stack that is published. So I will not spend a lot of time, but really high level, in our pipeline, what we're trying to do is trying to leverage this idea of variability. So if you have these cell type specific programs, means that maybe you're using this mark in different ways in each cell type. So in this panel here, imagine that the rows are the cell types. So what we do is like we assess a way that without calling peaks, what are the regions that are most variable for this mark, and then give us a way to focus in the genome, to say like these are the regions that are more dynamic in the genome, and then you can still go after cell type specific mechanisms and extract transcription factors and then using RNA to see if this factor are, indeed expressed in the cell type that, where we predict their activity. So this was, just one way that, we proposed. Many other groups have proposed ways to do this, but, this is like a potential tool that you might want to consider to highlight region. The other thing, like I mentioned before, highlight perturb. So now you have, a candidate set of regions. This can be based on, like chromatin marks, can be based on, SNPs from GWAS or, EQTL. But, gene editing technology has been around for a while. What made, CRISPR so like disruptive is that this system is so easy to program. And, if you're not familiar with this system, there are like two parts. So there is this Cas9 protein, and then there is this RNA that is called guide RNA, where, one part of this guide RNA essentially can be synthesized and can be directed to almost anywhere, any location in the genome. And, by simple complementary rules, you can essentially program this sequence and deploy and, go and essentially create double-strand break. So one, small thing is that, the fact that you can go almost anywhere is that there is something called PAM, that, this short sequence here that, you should match in order for this system to activate. But recently people have proposed actually a way to relax this constraint. So you can essentially perturb almost any region in the genome. And what happened, after you introduce this double-strand break? So there are like different repair pathways in the cell. Two common one is called non-homologous enjoining. They will create small insertional deletion. And the other one is called homologous direct repair, where if you provide a small donor template, so this pathway that is less efficient sometimes can essentially resolve this double-strand break by using this template. And then what happens is you can essentially introduce whatever modification you want in that region. But this is like way less efficient. So this one is more efficient, and this is a great tool to go there and do this genomic vandalism, as George Church sometimes referred to this. But it's really nice to disrupt, for example, transcription factor binding or SNPs. And there are also the nice things about this system is that people have evolved, this system in really creative way. For example, one way is that you can, for example, remove, this capability of cleave the DNA, and you can modify the Cas9, and this is called dead Cas9, so it cannot cleave anymore. And then you fuse other, modifiers that, for example, repress or activate, locally the genome. And these are called CRISPR-I or CRISPR-A, and there are like so many variants to of this enzyme. But the idea is that you can repress, for example, an enhancer, or you can create an enhancer, wherever you want in the genome. And so now, like I introduce one way to highlight, one way to perturb. So what can we understand with these technologies? So one thing that like I did as a postdoc, I was really lucky to collaborate with Daniel Bauer and Stuart Torking, and they are really like focusing trying to understand blood development diseases, and they are really interested in trying to find ways to cure sickle cell disease. So in 2013, we published this paper where they were studying this gene called BC11A, and in particular, there is an enhancer of this gene that is really cell type specific, and this gene has this function to repress fetal hemoglobin. So as you know, like we have different kind of hemoglobins. We have the one that is expressed at the fetal stage and the one that is expressed at the adult stage. So one thing that like has been observed, some people have variants, and like they still express the fetal hemoglobin at the the adult stage, and these people are like have better prognosis if they get sickle cell, because they have like a backup, like hemoglobin. So in this paper, we show that if you delete this enhancer for this gene that is a strong repressor for fetal hemoglobin, you can actually reactivate, fetal hemoglobin. And this is like, a potential way to cure this disease. So this was, done at that time with talent. CRISPR was still not so popular. So we chopped the entire, enhancer, and we showed that, this was a key element for this gene. In 2015, we made, the realization, like, do you really need, the full enhancer? Maybe, there are like specific parts of this enhancer that are important. So we designed one of the first CRISPR-Tiling screen, and I will present, the idea better. But the idea is like you can create several perturbations. You try to find the smallest, part that you really need. And, indeed, we found, in this paper that you really need a single binding site to shut down this enhancer. And this was a binding site for a factor called GATA1. And also there was another factor, TAL1, nearby. And then in 2019, there was this new technology called base editing. And, actually this was, another paper. But in this paper, we show that actually, if you take a single perturbation, this element, and you modify human hematopoietic stem cell, you can actually, when they differentiate, you have a way, like to essentially reactivate fetal hemoglobin. So the idea is like you may take cells from the patient, you modify, you check if you're modifying this hematopoietic stem cell right without off targets, and then you reinfuse. And the idea is like, when they repopulate your blood system, they should have this fetal hemoglobin active, right? So this idea that, seems like reasonable, was actually really translation. So there are like now several clinical trials, one, led by Daniel Bauer and also from companies. And this was, one of the first demonstration that, if you combine genetics, functional genomics, and these technologies, you can create therapies that, actually are working. So, it's really exciting, moment, like where, you see the combination of computational methods with, like experimental strategies. And now, like, this is like one of the first, but there are a lot of many more things that we can discover in the genome. So, and this brings me to this this idea of this CRISPR-Tiling screen. So you can think, like we have these new tools that are really like easy to deploy. So, if you want to discover, the part that are important in the genome, you can play, kind of battleship, right? So there are like few things that you want to discover, and now you can just deploy this perturbation. Sometimes, you hit the right spot. Sometimes you don't find anything. So, and, like the cool thing is that you can be smart, in this exploration, because different technology have different perturbation range. Some they perturb, like larger region, 1 to kb. Others they perturb, individual nucleotides. So one thing I proposed on my R35 was, what about if we combine these technologies where we start with this low resolution scanning, using CRISPR-I and CRISPR-A, and then we do like maybe Cas9 perturbation that, introduce, 10 base pair, elements, and then you can go finer resolution using base editing. So this is something that, we are currently like exploring, and, I want to present, some of our work on this front. And, CRISPR-Tiling screen has, have been already used like by not just our lab, but many labs are using this tiling screen. These are just 3 examples of this tiling screen. One is this BC11A, story I told you, where we used, the Cas9 perturbation, and we were, using this enhancer. The purpose was this enhancer dissection. And, at that time, we proposed a hidden Markov model to do that. And then Jesse Engritz and Eric Lander, I know Jesse will probably present also here, they did a CRISPR-I screen where the idea was to discover enhancers, for the MYC gene. And, they propose moving average and t-test. And then the Alex Marson lab, they use CRISPR-I with the idea to discover an answer or, potential candidate enhancer by reactivating them. And they use, this CRISPR-I system, and, they use a simple strategy. So if we go a little bit deeper, for the general idea of this CRISPR-Tiling screen, the way it works, in the design stage, you have to think about what I'm trying to perturb, right? I'm perturbing, like, for example, distal region for a gene, or you might want to perturb the coding region of a gene. So this essentially will dictate, what kind of perturbation you want to use and the scale. The experiment, the way it works, after you design your perturbation, you will infect your cell. You can do lentiviral transduction in a way that you want to have one perturbation per cell. Like that's the typical setup, and you can play with different way to obtain that. And after you do that, you can have like a phenotype that you might want to study. For example, can be cell viability, can be like, as I mentioned before, the reactivation of fetal hemoglobin, or can be anything that you can measure, with FACS or, with other technologies. But the key idea is that you can essentially create groups. The simplest setup, you create two groups, one that have your phenotype, you can call the I group, and one that doesn't have your phenotype, you call the log group. So now what you can do is like, you have these cells, right? The one that responded, the one that didn't respond, you sequence. And then you can essentially try to create a table where you read out which perturbation. So this sgRNA is the particular perturbation that you introduced. And then you can essentially count how many times this perturbation was in my I group, the one for my phenotype, how many times was in the log group. And given that you design, this perturbation, where they map in the genome, right? So you can, imagine that you map back this perturbation, maybe this will map here, this will map here. So you can kind of create a signal, right? And then based on the enrichment of this signal, you can discover which part was associated to your phenotype. And, like just to, provide a little bit more detail, right? And, if it's not clear, feel free to stop me, raise your hand and say, Luca, can you explain better? But, we're here, right? So we have a region, we introduce our CRISPR perturbation, we sort. So what do we sequence? Let me provide you a little bit more detail. So the way it works, where you,
Let me chase the guy.
Oh, okay. Oh, sorry, guys. Yeah, I will.
Oh, hold on, I'll go.
Yeah.
I'd have to tell
Maybe
the story without this.
Maybe you can try to memorize. Like just keep in your mind. So what you sequence, you introduce this, like this construct that contain the guide. You see this guide RNA that get transcribed, and then with the Cas9 will go in the set, in the target that will modify, and then there's this puro that is like something to select only the cell that have this perturbation. But the nice thing is that whatever you introduce apart from the guide is the same. So you can design primers around this. So you will read, you have a simple way to read out which perturbation you have. And then, like you can derive a score, and the simplest one can be just a log2 fold change of your count in the I population, the low population, right? That's the simplest thing that you can do. So that's essentially a tiling screen, right? And it's really simple setup. However, things are simple, but when you analyze them, there are always, problems. So, really high level, we are here, right? So we sequence, and these are, our FASTQ file where we, see the abundance of, the guide in different conditions. And then there are a lot of, challenges. What we want is something like this. So we want to have kind of a denoised version of our signal. So each kind of point is like one perturbation. So we want to denoise the signal, and this can be positive and negative. Things that are positive may increase your phenotype. Things that are negative are decreasing your phenotype. Then some kind of statistical test to say, well, if you're around here, this is noise. Maybe this is something that you should focus. So to do that, we developed this tool called CRISPR-Surf, and this was developed by a PhD student in my lab, Jonathan Su. And so the way we approached this problem was, we can think about this CRISPR perturbation as a convolution, and the convolution. And imagine that there is some underlying, function of the genome. You can envision that, if it's red, is increasing your phenotype. If it's blue, it's decreasing. So you have positive and negative signal. So when you try to infer, this functional association with your phenotype, you measure this with a tool that can be like a gene editing tool. And what you measure is not the way the truth. Is like the truth that is convolved by this editor. So, and we want to go back, and I will try to motivate this intuition a little bit more. So the first thing that we need to think about is that what is the actual resolution that we have? So the first thing is that if you have a Cas9, so as I mentioned before, you do double strand break, and then you do short indel, and resolution here can be around 7 base pair. So you cannot perturb a single nucleotide. If you use CRISPR-I, CRISPR-A, based on previous literature, here you may perturb 200 or 500 or sometimes even 1 kb. So that's the actual resolution that you have with this tool, and you don't modify the DNA. Okay. So now with this in mind, what's going on here? So imagine that you have a function, right, that you want to measure that is like the underlying genomic signal, right, associated with your phenotype. So if you will have the perfect tool, can be like just a stick, like a discrete delta Dirac, if you want to formalize this. So you can imagine you can slide this thing on this function, and you will get, assuming you have no noise and no distortion, you will reconstruct this, at the right resolution. But we don't have this, right? So we have, maybe something like this. So when we slide, imagine that you do a tiling screen in all the position, what you do is kind of convolving your signal, and you get a smoothed version of your signal. And if you have CRISPR-I, the perturbation is much larger. So this operation is called convolution, and people have been, studying this for several years, in image, in audio, and many other, application. So if you formalize this, you have a convolution operation where you have the genomic regulatory signal, the CRISPR, kernel, we can call that some noise, because we always have noise. And then this is what we observe. These are our observed data. So now can we, given the observed data, and given that the average profile, what is the most likely signal that you can, reconstruct? So there are so many ways to solve this, right?
Is the noise, is there different structures? Noise, what kind of noise is?
So the noise can be multiple things. One can be sequencing errors. Another thing can be potential, batch effects, because you need to do replicates. And then, another source of noise can be different sequencing depth that, may essentially not essentially allow you to measure all the guides all the time. Or maybe when you do this transfection, the transfection is not always the same. So some guides are more expressed than others. So there are a lot. So here I'm just hiding all these things in one term, but the noise, can capture some of this. So these are the things that we understand, but there may be other one that, I don't know. So the way we do this, there is one thing called generalized lasso. And so essentially that's, there are many more details, but just want to give you the intuition here. So So one thing that we do here is like y is like your observed signal, beta here this the coefficient you want to learn that are this genomic like association that you want to discover. In X, essentially we encode this perturbation profile plus the location, because as I mentioned before, so in reality X is like the product of two matrices, because you cannot perturb every single region, so we try to essentially account for that. And then in this generalized loss formulation, this D, like what we do, essentially there is one thing called fuse penalty that essentially try to, instead of like in the normal L1 where you say this beta cannot grow like too much, you would penalize with that. Here what we try to do, like we try to minimize the difference between consecutive beta, so you don't want these things to jump up and down too much. And the intuition behind that is that in an answer often you have blocks of regulatory element, regulatory region, like imagine a binding site, right? You have 10 to 20 base pair that are all like together. So with this fuse penalty, we're trying to, like, when we try to propose solution, we're trying to propose solution that have destruction, right? We don't want to have a nucleotide in the middle of nowhere. So, and in our experience, this seems to work well. So that's what we do. I mean, there are like more details here that are described in the paper, but this is the intuition of why we use this framework. So we tried this in multiple screen. The nice thing about this formalization, you can apply a different resolutions that we use in Cas9, in this CRISPR-I and CRISPR-I. So these are the three screens I had a slide before. The nice thing that we saw is that we recover all the validated elements that they have in the screen, but we find also new elements. We didn't test because, like we didn't have the system, but one cool thing that, we show in the paper is that the new element that we find are decorated with, chromatin marks or chromatin accessibility that suggest that, they may be like functional element. And, they were not essentially discovered with the previous methods because, they didn't have the power. And then, one cool thing that we did in collaboration with the Bauer lab was to do like a screen where we use two technologies to target the same elements at the same time to compare, the resolution. So this again was, this BC11A region, and we target multiple elements. And then, long story short, what we learned here is that so these are three elements that are both, significant based on our tool. For CRAB, this CRISPR-I, sorry, you have like much lower resolution as expected because the perturbation range is much larger. And if you compare the top one with the bottom one, Cas9, can really like, get, smaller part of, this enhancer. So, it's really nice, and also can potentially tag elements that have, this opposite effect. Whereas, with Cas9 that, you have just epigenetic perturbation, it's really hard to have, this dual, sign. The other thing that, is not surprising, but it's good to keep in mind if you want to do the screen, if you use epigenetic perturbation, you may not see any effect in coding regions. Whereas, if you perturb an exon with genetic perturbation, probably you will mess up, the protein badly, and you will see a strong effect. And then around the TSS, again, we see a similar trend. The Cas9 is a little bit more higher resolution. So we build, an interactive website, also a command line. And, the idea here is like, we want to make this simple so, people can design this perturbation, and, they can even fine tune depending on, future, editors. And then you have this kind of step-by-step, process where you can deconvolve the signal, and, below you see the raw signal, and then on top, what we reconstruct with this framework. And then there is like a statistical test that tells you like which region you should focus. And it's all interactive, but we also have a command line for people that do really large screen where maybe a website is not the appropriate tool. Thank you. And then, like where we go from here, right? So far, like I show you like screen based on Cas9 and CRISPR-8, but the genome editing field is going so fast. Yeah. So they are, like creating new tools like every, few months. So presented, these nucleases, but now we have these base editors that, they use nick case with, some deaminase, for example, domain, and, this can introduce precise substitution. A more recent tool from the David Liu lab is called prime editing. It's a really complex system where they use reverse transcriptase, to like introduce, essentially whatever you want in that region. Is also based on a nick case, Cas9. So with this editor, you can be a little bit more ambitious and, just really precisely try to understand small region, right? And, you can try to do saturation mutagenesis of really small part and really try to recover a more detailed version that, if you delete the element like with Cas9. So I presented this approach. One thing that we're exploring now is like, one thing that, like code name, CRISPR clear is not published. So here, the idea is that instead of, measuring the guide abundance, right, as I showed you before, what about we measure, like the alleles that we create? And then we try to essentially correlate the alleles to the phenotype. Because, like all the things I presented before, they measure, just the guide presence, right? So you don't actually know what is happening at that region. But if you actually design primers to to quantify what are the alleles, you may understand much better, like what is up. So, it's not anymore trying to resolve what each guide is doing, but you can actually measure. For this, we created this tool, and see I'm running out of time, so I will go a little bit faster. But this tool called CRISPR-2 is a tool to actually quantify genome editing activity. So, and you can use for any editors. So the key idea, here is that imagine you're modifying this region, you design primers, you amplify this region, and then you sequence. And, this is called usually amplicon sequencing. And then, this tool allows you, it's also a website, you can just drag and drop your FASTQ file. So create a really long plot and, report. And there are like many outputs. The two ones that are important for this exploration, one tells you like on average, how many edits do you have? Like how much are you, how efficient is your system? The other one is a table like this, that, is probably the best way to really see what is happening, what are you doing to your region? And this essentially show you the reference and then all the alleles that you create with their proportion, right? So now that you have these tables, what can you do? So you can start to compare, like population, right? So as I mentioned before, maybe you have a population of cells that respond to your phenotype, population that didn't respond to your phenotype. So by comparing the proportion of the alleles, you can try to reason about which part or which particular insertion, deletion, substitution are functional, and then try to think about, oh, maybe there is a binding site for a factor, but here you have this direct observation of the alleles. So it's not like guessing that maybe this is the region. So this is like we are building now like computational tools to take these tables and try to like have a nucleotide resolve signal where we can say this particular nucleotide has this effect. It's similar to what you do now with MP array, but like MP array is like much easier to do this. For this, you have to like build all these tools and create all the protocols. So, and this tool is available online. And now, like I'm really excited, as Alan mentioned, to be part of this new consortium where we don't go after anymore two elements, where we are trying to reason about individual variants. And I'm directing a center, called Characterization Center with several other PI, Daniel Bauer, Guillaume Lettre, Richard Sherwood, and some colleagues, Benjamin Kleinsteiber and Benur Lee, where we're using base editing to essentially perturb individual variants that people have essentially highlighted by GWAS or summary statistics, GWAS or QTL or other studies, and really try to understand, by perturbing them, like if they're really like, functional, right? So, we do like, instead of doing fine mapping with statistical methods, we just go there and just perturb all these variants and we just see which one is important. So I see it's like 4:45, so maybe I can try to wrap up in 5 minutes. Okay, I would be a little bit more quick. In 5 minutes, I just wanted to tell you, like, my lab is also really excited about single cell assays. And, these are all unpublished work, but, we have preprint for 3 of them. So one, like I will briefly present, is called SIMBA. We have a tool called Megatron that you can use to model lineage data, single cell expression data, where you have also lineage barcode. And we also work in velocity, proposing a probabilistic framework with uncertainty. And then also we are developing this method called dictis to reconstruct developmental network. So if you are curious, we will have time later. So in the last 5 minutes, I want to just give you like some ideas about this work called SIMBA. This is like a work led by Dong-Chen, postdoc in my lab, in collaboration with Adam Lehrer, Jiang Ryu, and Michael Vineyard. Adam is from Meta AI, like Facebook. So here, like, as you know, single cell omics, is really an exciting area. And we have assays that, allows you to measure in single cell expression of thousands of genes, chromatin accessibility, methylation, protein level, so many other things. This is also fast evolving. So it's a really exciting area. And the goal of these regulatory maps that, I mentioned of SIMBA is like, you take all this measurement and, we want to build these maps where, they look like, what you have now in many single cell paper. But the key difference here is that the dots are the cells, as, the normal UMAP, but you have also dots for the features. So you may have like, a dot for genes, motifs, and peaks. So you can essentially reason about, the function or the relationship between different features, depending on where they are in these maps. So for example, you can say, if you're in this cell state, these factors are important, and, these factors are close to these peaks, and these peaks are close to this gene. So maybe they form, regulatory circuits. So that's the idea of these regulatory maps. Given that I don't have time, I will just skip some slides. So the way we are building this regulatory map is using some technique borrowed from natural language processing, where, you have language, you have text, and you want to teach like a machine to understand text. And the idea there is that you can take word or sentences and you associate vectors and you create spaces where, the relationship, the distance between words may, like represent some semantic or some, for example, one, classic things is like, if you use this word to vector technique, to create a queen, you take the king vector and then you just add woman minus man, and you just get the queen. So these, techniques are really powerful, and, these embedding spaces are really like, you can learn a lot about the context of so the idea is like, can we do the same with, like sequences and, like measurement from single cell? So the idea is like, can we do this kind of maybe chromatin tweets where, a peak can tweet and say, hey, I'm present in these cells and these are the motifs. So the way, we do this, we use a technique called graph embedding. So I will skip some slide. So and maybe I would just show you this and maybe one vignette and then we can end. So the idea is like, you take all the single cell measurement that you have, and then you try, instead of doing the classic things where you do clustering or, try to align these matrices, you just build a graph. of all the measurement that you have. So in this graph, you have the cells that you measure, and then you can say, if you have a gene expressed, you create an edge. If you have a peak, you create an edge. If a peak has a particular TF or chimer, you create an edge. So this graph, as it is not really helpful. So what you do, like you embed this graph. So what you do is like you create a vector for each of your node, and then you create this space, and this space is the property that you have the cells, but you have also all the other features that are created. And then by reasoning on the proximity of this, we show, like, and there is a preprint, but also happy to talk with you. We show that you can essentially find, without doing any clustering, important marker for the population. You can find relevant transcription factors, and as well, regulons. And all of this is based on the proximity of the points in the space. So there is no clustering, there is no other, ad hoc procedures. And just show you this maybe vignette here, a single one, where this is like a data set where we have multimodal data. So you have gene expression and chromatin accessibility measured in the same cell. So the graph looks like this, where you have the cell connected to genes, cell connected to peaks, peaks connected to chimers or TF. So we apply this procedure, and then you can build these maps where you can embed just the cells, and you can just do the clustering if you want, like using standard tools. You can have cells and genes, or you can have cell, genes, motif, and peaks, all in the same map. And then we have, a way using this technique to find the important features that define the space without the clustering. And then based on this procedure, then we can reason about, like, for example, you find the most important TF. Then you can ask, what are the relevant cell states for this TF? What are the peaks where this TF, might be important? And, just to end, just to show one important, point, powerful, approach that you can use in a few of these maps. For example, in these maps, you have embedding for genes and motif, transcription factor motif. So you can, for example, sort all the transcription factor based on the distance between the embedding of one factor and the motif. If you rank all of them, you will find all the master regulators for the system. And, like, this is just a simple proximity operation. And then, you can reason about peaks and genes nearby, you can find the regulon. So we show that this can be also applied for, batch correction or, other tasks. I think it's a new way to think about single cell modeling, and I'm excited, to see where this will go, but I'm happy to chat about this. So I will skip the conclusion. So I just wanted to acknowledge my lab and, Adam from Meta AI that, really helped us with this graph embedding, and Michael and Jiang that helped in this project, all the lab member and my funding sources. And I will stop here with that question. Thank you. Thank you.
Any questions?
Yeah, really nice talk. Curious about, for some of the tiling, I guess the nuclease kind of conventional CRISPR tiling screens. I think different groups have found an issue where guides that are, a little bit proximal to the start of an exon can make these larger deletions.
Yes.
And then those have usually much stronger effects than, ones that perturb an enhancer or something. So is there a way to correct for that in this kind of model? Or can you get it from the sequencing, was it the clear approach that you showed?
Like in the model that we propose, we don't correct for that, because we've been focusing more on an answer element. So this is something I didn't explore. But yeah, maybe like by reading the alleles, I think would be much easier to correct for that. You know, the count base you don't see, right? So you have to then use predictive models and, you know, assume that you may have longer deletions. So I don't think it's easy, but for the allele base, I think, you know, there are potential ways to correct for that. But, you know, it's not included in this current framework.
And how good are the predictive models? I know different groups have developed models that will predict the NHEJ or the, you know, the MMEJ outcomes of individual guides based on the PAM -4. Are those predictive enough to be useful in this kind of a model?
Yeah, I mean, it depends. You know, we played a little bit with that. One thing is that this model was trained in particular cell types. So if you have a cell type that, you know, is similar, probably they are more effective. And also we observe, you know, sometimes, you know, for the top allele, you know, like actually they seems to work well. The order may be off, right? But, you know, if you just ignore the actual frequency, you know, you take, you know, the top 5, usually you find things that, you know, the model would predict. But if you want to reason about the actual editing, this is something that I don't think, you know, they are, you know, yet there, right? Maybe we need a little bit more data. But if you want to reason about potential outcomes, I think, you know, they are effective.
Okay, yeah, great. Very cool.
Also, if there's any questions online. Okay. Any questions?
Maybe I can.
Any other questions here? So I've got a question. In the convolution deconvolution part of your talk, did that give you any insights into ways that you can improve the DCAS assays like CRISPR-I, CRISPR-A, in terms of maybe density of tiling or increased sequence depth? What are the parameters that improve your ability to deconvolute?
Yeah, actually, yeah, in the paper, we actually investigated this a little bit. So we did like some downsampling. And so if you use CRISPR-I, actually, you don't need many guys. They are like, you know, these things are, you know, so powerful. And so I think, you know, like if I remember correctly, you know, what we were suggesting is like every 30 base pair probably is good enough. And then, you know, sequencing is important, but it's not, you know, I think, you know, if you are like aim to have like a representation of 300 per guide, you know, probably it's okay. For Cas9, for base editing, I think, you know, you need to like tile, you know, much denser. So the denser you go, like the better. And then when we did this, you know, we were using NGG. So, you know, you have like some moles where, you know, there is not much you can do. But now, like, you know, Benkley Stiver that, you know, is in our center, proposed this SPRY and, you know, SPG where you can essentially tile most everywhere, but you have less efficiency, a little bit less. So, you know, just a compromise. So yeah, what we learned is like sequencing depth is not super important for count base. For the allele base, you know, we're investigating this now is really important because you may want to go after rare alleles that can be still really like associated with your phenotype. But for count base, you know, you don't have the resolution to go after those things, right? You just say, yeah, there is something here, but you don't know which nucleotide. Yeah.
Okay, thank you. Other questions? Okay, let's thank Luca again for. Thanks.